Hey folks, it's me again, Mr. Lang, your friendly neighborhood biology teacher, and today I want to take you on a journey through the Calvin cycle to end photosynthesis and wrap things up with that sugar production. That's how we make glucose. So just to go back to our chocolate factory analogy real quickly, sort of like we got a powered power plant over here. It's going to send energy to the Calvin cycle to make the sugar. And right now, I want you to stop for a second and think to yourself, what is the energy source that the light-dependent reactions send over to the Calvin cycle to run the Calvin cycle? If you said NADPH and ATP, you are right. Ding, ding, ding. Give yourself a gold star. And now it's time to dive into that Calvin cycle. If my cursor will work, there we go. So realize that in the Calvin cycle, we're basically taking carbon dioxide from the atmosphere, and through a bunch of reactions, we are going to reduce it to glucose. And the way that that's going to happen is through this process called the Calvin cycle. And in the end, we're going to end up with this molecule that has a lot of stored chemical energy because it's pretty fully reduced. In that reduction from CO2 to glucose happens in a lot of small steps, and each of those steps is catalyzed by specific enzymes, which we're not going to get into except for one of them. And at every step, we're going to use energy from the light-dependent reactions, either ATP or NADPH, in order to drive the reactions. Where is it happening? Well, it's happening in the chloroplast, like everything else with photosynthesis, but it's not happening anywhere near, well, sorry, it is happening near the thylakoid, but it's not happening in the thylakoid or on the thylakoid membrane like the light-dependent reactions. This is happening in the space, the fluid-filled space in between the thylakoids, those grana are the stacks of thylakoids, so it's happening in this sort of space in between the thylakoids called the stroma. That's where the Calvin cycle is happening. So here's the Calvin cycle. And again, like everything else with photosynthesis, this looks daunting when you first look at it. I'm going to tell you what you need to know and what you don't need to know, and I'm going to explain anything that you need explained. So don't worry about it. Let's just talk about it. So we're going to start with our input. We're going to have carbon dioxide added. So carbon dioxide is going to come in, and there's an enzyme here called Rubisco. I know it doesn't end in ASE, so I'm making a liar out of myself because in one of these previous videos I said enzymes always end in ASE and they have something to do, their name has something to do with what they do. Well, Rubisco doesn't really follow that trend. So Rubisco is an enzyme that is going to take CO2 and combine it with RUBP. And what that does is it's basically going to shuffle around a whole bunch of carbons. And if you look here, you'll see this little number in front of all these. So this is 3 RUBPs, and this is 6 phosphoglycerates. And this is 3 biphosphoglycerates. This is 3 carbon dioxides. So it's important to keep those numbers in mind, and I actually have a more elemental version of this as the next slide that you can see, and it shows all of the molecules at the same time. But anyhow, Rubisco is going to combine carbon dioxide and RUBP, and it's going to sort of shuffle up the carbons into 6 3-carbon compounds over the course of a bunch of turns of this cycle. And we're going to add, there's that one, we're going to add a whole bunch of ATP, we're going to add a whole bunch of NADPH, and the College Board doesn't necessarily care that you know about the intermediary steps, but it does want you to know that carbon dioxide and RUBP are combined using Rubisco, that a bunch of ATP and NADPH are used, and that you end up with 6 molecules of G3P. Now, that G3P is also known as PGAL, so you'll see it referred to as that in some places, but often G3P. 1 molecule of G3P basically equals half of a glucose. So we get 6 of those. 1 of them is siphoned off to create glucose, and 5 of them go back into something that sometimes is called the carbohydrate shuffle. And we take this G3P and we will use it along with ATP to recreate the RUBP molecule that's going to be used as by Rubisco to fix carbon. And that's the term that we use. It's carbon fixation. All that means is we're taking carbon that's in sort of a gaseous form and we're fixing it to something larger, something solid. So we're fixing carbon in this whole step here. Also important to note that glucose is one of the things that can be made by G3P, but also G3P is a pretty common precursor for all sorts of other things. So common precursor for fatty acids, amino acids, all sorts of different things. So, so keep that in mind because it's important. Like I said, I have another diagram and it flashed up a second ago that maybe makes it a little more conceptual because you can actually see how the molecules are being reshuffled. So here we've got 15 carbons here. We've got 18 carbons because we added 3 carbons. So you can sort of follow the addition of the carbons to the molecules. And then we siphon off one of the G3Ps and 5 of them go in and get reshuffled again. And when we get rid of that, one G3P, we're back to 15 carbons total. Here we have those 15 carbons total. So you can take a second to look at this. You can pause the video, try and make it make sense for yourself. We can also talk about it in class. But try and make it make sense to yourself right now, because if you spend a little time with it now, questions will arise and you'll be able to better verbalize your ideas. So Rubisco, it's a really interesting enzyme because it is responsible for carbon fixation and is present in everything that photosynthesizes. It is probably the most important enzyme in the world because without it, we couldn't have autotrophs making glucose for us to consume and make into ATP in our bodies. Basically, it builds life out of the air, which is incredible. It's probably, well, definitely the most abundant enzyme, but one of the reasons it has to be so abundant is that it is also one of the least efficient enzymes that there is. It only catalyzes a few reactions per second, which on the molecular scale is slower than molasses, as my father would say. And you have to think about the idea that most enzymes catalyze thousands of reactions per second. This enzyme, Rubisco, catalyzes a couple, 3, 4 per second, which is really, really slow. But, you know, it's really important too. So thanks, Rubisco. So overall, the Calvin cycle, we've got this G3P PGAL molecule that comes out. 6 turns of the overall cycle is going to equal 1 glucose molecule. We can do the math and show how that works if you'd like to. And then again, it's really important to realize that G3P is not just the precursor for glucose, which can then be converted into other carbohydrates, but it's also a precursor for lipids, amino acids, nucleic acids. It's really a precursor for a lot of the biological molecules that we need. So in summary, if we wanted to think about the overall things that we've been talking about from the original overview video through the Calvin cycle, we've got our light reaction. Our light reactions made ATP and NADPH, used water, and it produced oxygen as a byproduct. The ATP and the NADPH were shuttled over to the Calvin cycle, where we consume CO2, we produce PGAL or G3P, we regenerated, which is kind of a weird way to say it, I guess. We used ATP and turned it back into ADP, and we used NADPH and turned it back into NADP+, which will now go back to the light reactions to get sort of recharged to be used again in the Calvin cycle. Lots of factors affect photosynthesis. Because enzymes are really important to the entire process, they drive the whole thing. That means that anything that affects enzymes will affect the rate of photosynthesis. So, for example, as the temperature gets colder, enzymatic action tends to slow down. Also, raising or lowering the pH can drastically affect the rate of photosynthesis or any enzyme-driven process. It's also important to realize that the amount and type of light can affect the rate. So if you have lots of cloudy days, you're not going to have as much photosynthesis going on. If you have a shortage of any of the reactants in either of the in the equation, or water, that's going to affect the rate of photosynthesis as well, because you can't have a process happen without having any of the reactants that are going to go into making the products. And photosynthesis, if we didn't have it, we couldn't feed ourselves. There's no way that we would be able to make enough energy without photosynthesis to create life as we know it on Earth. So it's a very, very, very important process. And it's important to understand how it works, because since this course is an in-depth look at life in general, photosynthesis is the process that life takes its roots from. We can't have life without photosynthesis. That glucose is of paramount importance. So the importance of the process can't be overstated. I know it may seem like I'm overstating it right now, but it really can't be overstated. So so it's a really important process. So some fun facts here. Each year, photosynthesis synthesizes 160 billion tons of carbohydrate. That's a billion with a B. That's a lot of carbohydrates. And as we know from the first presentation, heterotrophs are dependent on plants as a food source for fuel and raw materials. We can't do it without them. So I'm going to leave you with just a quick look at a really simplified overall version of the energy cycle, where we've got photosynthesis, getting energy from the sun, creating glucose and oxygen. Glucose and oxygen go into cellular respiration, creating CO2 and water, which are the reactants that go into photosynthesis. It's a really great cycle. I know that the first presentation that you saw, I started with the actual equations for these two things, but I thought that this was a good place to end. That's why I thought this was a good place to end, because it sort of brings it all back together and brings it all back home. So if you have questions, make sure you ask them either via email or in class. And I hope this helps. Let me know if you have any problems. Have a good day.